Hey fellow lab rats, this is Rebecca from the Lab Rat YouTube channel. In this video we're going to be discussing connective tissue and muscle tissue staining in the histopathology laboratory. Alright, let's get started. There are four basic types of tissues. Those are connective tissue, epithelial tissue, muscle tissue, and nervous tissue. This lecture is going to be discussing the staining of two of these basic tissue types, connective tissue and muscle tissue. So connective tissue helps to provide both structural and metabolic support. Connective tissue consists of three different components, including fibers, cells, and amorphous ground substance. Additionally, there are four different types of connective tissues. These are connective tissue proper, cartilage, bone, and blood. Special stains are used for the demonstration of the fibers and the cells of connective tissues. We'll get to these special stains later on in this lecture, but we first need to discuss the fibers and the cells. So the fibers of connective tissue include collagen fibers, elastic fibers, and reticular fibers. The cells in connective tissue include fibroblasts, mesenchymal cells, adipose cells, mast cells, macrophages, plasma cells, and blood cells. So let's briefly talk about these fibers and cells. Collagen fibers function to provide strength to the tissue. An arrangement of collagen fibers are present in tendons, dermal tissue, and organ capsules. There are 7 types of collagen fibers, with type 1 being the most common type found within the human body. We can see collagen fibers with a light microscope. They are birefringent under polarizing microscopy, and they have a cross striation pattern in electron microscopy. In the histopathology laboratory, collagen fibers are demonstrated with the Masson and Gamori technique and with the Van Giesen stain. All of these stains will be discussed later on within this lecture, so don't worry if you don't know what they are now. Elastic fibers are in fibrous connective tissue, but are most frequently found in tissue that needs to be flexible. So elastic fibers allow tissues to stretch. They cannot be seen on regular H&E stains. They are only visible with Verhoff, Weigert, and Gamori stains. Again, we'll discuss these stains later on in the lecture. Reticular fibers are a type of collagen. They cannot be seen with regular light microscopy, but can be visible with an argyrophilic reaction. And this term means that they are able to bind with silver ions. And once those silver ions are bound with the reticular fibers, they are chemically reduced to their metallic form, which is visible. So those are the 3 fiber types of connective tissue. So let's talk about the cells of connective tissue. So fibroblasts are the most common cell type present within connective tissue. They are responsible for producing the connective tissue fibers. These are not visible within the tissues. So the photo on the right-hand side of the slide here shows fibroblasts that are just growing in a cell culture. Mesenchymal cells are cells that are undifferentiated, meaning they don't know what cells they're going to become yet. If a specific cell type is needed by the body, these cells can develop into differentiated cells, meaning they decide what cell line they will become. Mesenchymal cells cannot be distinguished between fibroblasts. Adipose or fat cells are responsible for both producing and storing lipids. So these are commonly found in loose connective tissue. Adipose cells are not preserved in paraffin sections. Now, special techniques can be used to help preserve them in frozen sections, and we'll discuss this later on within this lecture. Mast cells are present in connective tissue and contain secretory granules that contain both histamine and heparin. They're usually present along blood vessels. Macrophages or macrophages are scavenger cells that are found both in connective tissue proper and also in the liver, myeloid, and lymphatic tissues. Now, monocytes are the precursor to macrophages. So these are present within the peripheral bloodstream. So when I say peripheral bloodstream, I mean like when you go and you get your blood drawn by a phlebotomist or a nurse, the blood that's being taken out of your body, that is from the peripheral bloodstream. So monocytes are in there and then become macrophages when they go into the tissue. So both monocytes and macrophages are not cells that are demonstrated in the routine histopathology laboratory. Now, plasma cells are derived from B cell lymphocytes and produce immunoglobulins, which are antibodies. So examples of plasma cells can be seen on the right-hand side of the slide here. Immunoenzyme techniques are now used to demonstrate immunoglobulins, but previously methyl green pyronin stain was used to help diagnose immunoblastic sarcoma cancers. Blood cells are also considered connective tissue, so these include red and white blood cells and plasma. Blood cells aren't typically discussed in histopathology laboratories as these fall under the medical laboratory technology profession. The basement membrane, or the basal lamina, separates the epithelial cell layer from the underlying connective tissue. The basement membrane is made up of epithelial cells, type 4 collagen, laminin, and a proteoglycan that contains heparan sulfate. It serves as a support structure for the epithelium as well as for cell attachment. I created this clip art here on the right hand side of the screen showing the location of the basement membrane in relation to both the epithelial cells and the connective tissue. So here are the epithelial cells, here's that basement membrane, the connective tissue here, and then nerves run through it as well. Hopefully that helps you give an idea of what we're talking about. So we've discussed connective tissue and the basement membrane. Now let's talk about muscle. So just like connective tissue, muscle tissue is one of the 4 basic types of tissues. There are 3 different types of muscle tissue: cardiac muscle, smooth muscle, and skeletal muscle. I've created this chart to show you the main features of each of these muscle types. Hopefully it helps to help you organize this material. So cardiac muscle is located within the heart and functions to help the heart pump blood. These muscles operate involuntarily. So what does that mean? So the heart muscles contract without you being aware of it. So you and I don't consciously think or focus on pumping our heart, right? So we're not like, okay, pump, all right, pump, pump. We don't think that. It just automatically happens, right? So this is an involuntary contraction. So cardiac muscle is striated, and this term means that the tissue has transverse dark and light bands throughout it. Each fiber has 1 nuclei present in it. Smooth muscle is another involuntary muscle that helps to move internal organs. It's present in the walls of blood vessels, internal organs, and also the gastrointestinal or GI tract. These muscles are non-striated, are spindle-shaped, and have 1 nuclei per fiber. Skeletal muscle is attached to bones via tendons and functions to move these bones for a source of heat for the body and also for maintaining body posture. So these are voluntary muscles. So we don't have to think about making our heart pump, correct? Right? So that's involuntary. But if we want to go take a walk, we have to consciously think about moving our legs in order for these muscles to contract to allow us to walk, right? So that's voluntary versus involuntary. Skeletal muscle is striated and tubular in shape, and they have multiple nuclei present in each fiber. So let's look at some photos of what these muscle types look like. This photo on the left-hand side of the slide here is an example of a cardiac muscle longitudinally. So the fibers are branched, and there is 1 nucleus that is centrally located per fiber. There are striations, but not as pronounced as they are in skeletal muscle tissue. The photo on the right-hand side is a section of smooth muscle. So the tissue on the left top part here of the photo, so like in this area here, okay? So here's the top left and here's the bottom right. I'm going to talk about these both individually. So the again, the left hand upper top left is a in cross, and then also the bottom right part of the photo is the tissue longitudinal, and this is a characteristic of external muscle layers of tissue in the gastrointestinal tract of humans. Now the arrow, this black arrow here, okay, is pointing towards a collection of nerve cells, which is also called a ganglion. This photo shows an example of skeletal muscle in a longitudinal section. You can clearly see the striated appearance here. Each muscle cell contains more than 1 nuclei. Now let's learn about the special staining techniques. So these are the Masson trichrome stain, Gamori one-step trichrome stain, Van Giesen picric acid acid fusion stain, Verhoef elastic stain, aldehyde fusion elastic stain, and the Russell modification of the Movat penachrome stain. So with all of these stains, we're going to discuss the purpose and function of the stain, what the tissue needs to be fixed in, how thick the tissue needs to be cut, what quality control tissues are indicated for each stain, the reagents needed, and of course the procedure for performing the stain. So the first stain that we are going to be discussing is the Masson trichrome stain. It's called trichrome because it uses 3 different dyes, so tri-3. So the Masson trichrome stain is used to determine between collagen tissue and smooth muscle tissue within tumors. It is also used to help identify if there are increases of collagen tissue in certain diseases. So an example of this would be cirrhosis of the liver. For this stain, tissues need to be fixed with Bouin solution. Of course, this is the preferred fixative, but 10% neutral buffered formalin is also considered acceptable. Paraffin tissue sections need to be cut at 4 to 5 microns in thickness. Now, most tissues have an internal control. However, if an additional control tissue is needed, you can use uterus, small intestinal tissue, appendix tissue, or fallopian tube tissue for this stain. The list of reagents needed to perform the Masson trichrome stain are Bouin solution, which is prepared by mixing 75 milliliters of picric acid, 25 milliliters of 37 to 40% formaldehyde, and 5 milliliters of glacial acetic acid. Weigert iron hematoxylin, Beibricht scarlet acid fusion solution, which is prepared by combining 360 milliliters of that 1% Beibricht scarlet with 40 milliliters of 1% acid fusion with 4 milliliters of glacial acetic acid. Then you'll also need a I'm going to butcher this, forgive me, a phosphomolybdic and phosphotungstic acid solution. So you want to combine 25 grams each of those different types of acid with 1,000 milliliters of distilled water. An aniline blue solution, which is prepared by combining 25 grams of aniline blue with 20 milliliters of glacial acetic acid and 1,000 milliliters of distilled water. And then lastly, a 1% acetic acid solution, which is prepared by combining 1 milliliter of glacial acetic acid and 99 milliliters of distilled water. So those are all the reagents that you'll need. So the Masson trichrome has a conventional procedure and a microwave procedure. To perform the conventional procedure, the tissue must be deparaffinized and hydrated to distilled water, then rinsed well in distilled water. The tissue then needs to be mordanted with Bouin solution. This needs to be performed at 56 degrees Celsius for a 1-hour period. And after that 1-hour period, the tissue needs to be removed from the oven, cooled, and then washed in running water until the yellow color disappears from it. And once that yellow color disappears, the tissue section then needs to be rinsed in distilled water. Then the sections need to be stained with the Weigert hematoxylin for a 10-minute period. After this step, rinse with running water for 10 minutes, then rinse with distilled water. Then the tissue slides then need to be stained with this Beibricht scarlet acid fusion for a 2-minute period, then rinse with distilled water again. After the distilled water rinse, the tissue slides need to be placed in this phosphomolybdic and phosphotungstic acid for 10 to 15 minutes. This solution then needs to be discarded after use. After this, the sections need to be stained with the aniline blue solution for 5 minutes, and then, you guessed it, rinsed in distilled water solution again. After this distilled water rinse, the slide should be placed in 1% acetic acid solution for a 3 to 5 minute period. Then dehydrate with 95% and absolute alcohol using 2 changes each of those. The tissue then needs to be cleared in 2 to 3 changes of xylene and then mounted with synthetic resin. When stained with the Masson trichrome stain using the conventional procedure, nuclei will be black in color. Cytoplasm, keratin, and muscle fibers, if present, will be red, and collagen and mucin will be blue within that tissue. When collagen is predominant within the tissue section, it is better to counterstain it with light green instead of the aniline blue counterstain. If a tissue section is stained with methylene trichrome and there is a decreased red staining, this means that the staining solution is too old and needs to be discarded, and a new staining solution needs to be utilized. Now, if the blue color and connective tissue is faded with this stain, this likely indicates that it has been over-differentiated with the acetic acid solution. If using the methylene trichrome microwave procedure, the tissue needs to be first deparaffinized and hydrated to distilled water. Recall that the fixative for this stain is Buin solution. If the tissue has not been fixed in Buin solution already, it will need to be mordanted with the Buin solution. So to do this, the section should be placed in a Copeland jar filled with Buin solution with a loose lid. So it then needs to be microwaved at 70% power for 45 seconds. If the tissue is already fixed in Buin solution, obviously the step can be omitted. So then the tissue section needs to be rinsed in running water until it has no color to it. It then needs to be placed in gilhematoxylin 3 in a plastic Copeland jar with a loose lid. And this Copeland jar then needs to be microwaved on high for 15 seconds and then rinsed with tap water. The next step is to blue the nuclei with alkaline bluing solution, followed by a tap water rinse, then a distilled water rinse. And after these two rinses, the sections need to be placed in Biebrich scarlet acid fusion in a plastic Copeland jar with a loose lid. The Copeland jar and contents are then microwaved at 70% power for 30 seconds. After this step, the tissue sections need to be rinsed in distilled water. Next, the sections need to be placed in that phosphomolybdic and phosphotungstic acid solution in a Copeland jar with a loose lid. This needs to be microwaved on full power for 15 seconds, and directly following, they need to be placed in an aniline blue solution Copeland jar with a loose lid. This Copeland jar and contents need to be microwaved at 70% power for 30 seconds. And after this 30-second period, the tissue slides then need to be rinsed in distilled water. Following this rinse, the section should be placed in 1% acetic acid for 2 to 3 minutes. Then they should be dehydrated with 95% and absolute alcohol, 2 changes each, then cleared with xylene mounted with synthetic resin. Now let's look at what the stain looks like. So the photo on the left-hand side of the screen is a methylene trichrome section of a vein. The lumen is seen visible at the top. So the lumen is a hollow passageway where the blood flows through the vein. So this lumen is this like white hole here, visible here. Okay. So the bright red area in this left-hand side photo is the smooth muscle cell of the vein, and the blue color is the collagen fibers that are present within the vein. Now the photo on the right hand side of the slide is a methylene trichrome stained liver section, which shows the portal vein. A large vein like this is good for judging the quality of the methylene trichrome stain. If the epithelial cells show red staining but the smooth muscle cells don't, then there is an issue with the stain and it should be redone. The photo on the left-hand side of the slide is a methylene trichrome stain of skin. So you'll notice that there is a green color and not a blue color, right? So this is because it has been used as, this light green has been used as a counter stain instead of the aniline blue. So the collagen is stained green in the skin section. Now the photo on the right hand side of the slide is a fallopian tube section that is stained with the methylene trichrome stain. So this photo on the right hand side is a pore stain, and you can tell this because the smooth muscle and the epithelium are not stained red, but you can see red cells that are stained red here. So red blood cells should never be used to assess the quality of a stain. So this stain is bad and needs to be repeated using new reagents. So all of these are the red cells here. But you see there's no other red here. This is a bad methylene trichrome stain. The Gamori one-step trichrome stain is a stain that is used to identify increases of the collagenous connective tissues in a tissue section, or to differentiate between smooth muscle and collagen fibers in a tissue section. In this staining procedure, chromotrope 2R, which is a plasma stain, and fast green, light green, or aniline blue, which are connective tissue fiber stains, are combined with phosphotungic acid. So this stain colors collagen either blue or green, and of course this depends on whether light or fast green or aniline blue is used. Paraffin tissue sections need to be cut from 4 to 5 microns in thickness, and most tissues have internal controls for this stain, so an outside quality control sample is not indicated. However, if desired, tissue from the uterus, small intestine, appendix, or fallopian tube tissue can be used as a quality control to ensure that the stain is working appropriately. The reagents needed for the Gamori one-step trichrome stain are Buin solution, Weigand iron hematoxylin, both of which we have previously discussed within our histopathology lectures on how to prepare. Gamori trichrome stain is also needed. This stain is prepared by combining 0.6 grams of chromotrope 2R, 0.3 grams of fast green FCF or aniline blue, 0.8 grams of phosphotungic acid, and 1 milliliter of glacial acetic acid, and additionally 100 milliliters of distilled water. So all that gets combined for that. Lastly, a 0.5% acetic acid solution is also needed. This is prepared by combining 0.5 milliliters of glacial acetic acid and 99.5 milliliters of distilled water. To perform the Gamori one-step trichrome stain, the tissue section needs to be deparaffinized, hydrated in distilled water, then rinsed well in distilled water. The tissue then needs to be mordanted in Buin solution for an hour at 56 degrees Celsius within the oven. The slides then need to be removed from the oven, cooled, and then washed in running water until the yellow coloring disappears from the section. After the yellow coloring disappears, the tissue needs to be rinsed in distilled water, then stained with Weigand hematoxylin for a 10-minute period. After the Weigand hematoxylin step, the tissue should be washed in running water for 10 minutes. Then the tissue should be stained in a Gamori trichrome stain for 15 to 20 minutes. After that 15 to 20 minute period, it should be differentiated with 0.5% acetic acid for 2 minutes, then dehydrated, cleared, and mounted with synthetic resin. When a tissue has been stained with the Gamori one-step trichrome stain, nuclei will be black in color. Cytoplasm, keratin, and muscle fibers will be red, and collagen and mucus will be green or blue in color. The photo on the right-hand side of the slide shows a large vein section that is stained with the Gamori one-step trichrome stain. Instead of using the aniline blue, light green was used. In this stain, you can see that the bright red color here is the smooth muscle present, and the green color is the collagen fibers that are present within the tissue section. The Van Giesen picric acid fusion stain is a primary connective tissue stain. However, it usually isn't used for this purpose. It's used most frequently as a counter stain in other staining methods like the Verhoef elastic technique. When serving as a counter stain for this technique, it is commonly referred to as the Verhoef Van Giesen stain. When in acidic pH solution, collagen fibers are stained by acid fusion. Picric acid in this stain ensures that there is an acidic pH. If the picric acid is not saturated, the collagen fibers will stain a pale pink to pale orange, and collagen, cytoplasm, and muscle stain all the same color. Any fixative can be used for this stain, and the paraffin sections need to be cut 4 to 5 microns in thickness. Most of all tissues will have an internal control for this, but if indicated, tissues from the small intestine, uterus, appendix, or fallopian tubes are good quality control tissues for this particular stain. The reagents needed to perform the Van Giesen picric acid fusion stain are Weigand iron hematoxylin, which we've talked about before, 1% acid fusion, which is prepared by combining a gram of acid fusion and 100 milliliters of distilled water, picric acid solution, which is prepared by combining 13 grams of that picric acid with 1,000 milliliters of distilled water. And then lastly, you'll need Van Giesen solution, which is prepared by combining 5 milliliters of 1% acid fusion solution with 95 milliliters of picric acid solution. To perform the Van Giesen picric acid fusion stain, the tissue needs to be deparaffinized and hydrated to distilled water. The tissue section then needs to be stained with Weigert iron hematoxylin for a 10 to 20 minute period. It is important that these sections need to be overstained, and the reason for this is because the picric acid will slightly decolorize the tissue, so it needs to start off by being overstained. So after this step, it needs to be washed in running tap water for a 10-minute period, then stained in Van Giesen stain for 5 minutes. This staining solution needs to be thrown away after use. Then the tissue slides need to be placed in 95% alcohol, then dehydrated, cleared with xylene, and then mounted with a synthetic mounting medium. When stained with the Van Giesen picric acid fusion stain, nuclei will be black in color. Collagen will be a brilliant red in color, and muscle and cytoplasm will be yellow. It's important that an iron hematoxylin solution be used because it is more resistant than aluminum hematoxylin to decolorization. If there is no sharp color differentiation between collagen fibers and muscle fibers when using this stain, this means that the saturated picric acid solution should be checked for viability. And an acidic pH solution is vital for this stain. This photo shows a large vein section that has been stained with the Van Giesen stain. The yellow colored part of this tissue here are the smooth muscle cells. The red color is, of course, the collagen fiber. The Virchow elastic stain is a stain that is used to show changes in elastic fibers. There are many pathological changes in elastic fibers, including atrophy of the elastic tissues, any arteriosclerotic changes of the tissue, and breaks that are caused by various vascular diseases. So in this stain, the tissue section is overstained with hematoxylin ferric chloride iodine. So it's overstained and then differentiated, so it's considered a regressive stain. Any fixative can be used for this stain, but neutral buffered formalin or Zenker solution are the preferred fixatives. The paraffin tissue sections need to be cut between 4 to 5 microns in thickness. For quality control for the Virchow elastic stain, aorta on its edge or a cross-section of a large artery can be used. The following reagents are needed for the Virchow elastic stain. You'll need Lugol iodine, which is prepared by combining 10 grams of iodine with 20 grams of potassium iodide, followed by 1,000 milliliters of distilled water. 10% ferric chloride, which is prepared by combining 50 grams of ferric chloride with 500 milliliters of distilled water. 5% alcoholic hematoxylin, which is the combination of 5 grams hematoxylin with 100 milliliters of 95% alcohol. Virchow elastic stain, which is prepared by combining 30 milliliters of 5% alcoholic hematoxylin, 12 milliliters of 10% ferric chloride, and 12 milliliters of Lugol iodine. Then you'll need a Van Giesen solution, and then 5% sodium thiosulfate, or thiosulfate, goodness, forgive me. So thiosulfate. That is prepared by combining 50 grams of the sodium thiosulfate with 1,000 milliliters of distilled water. To perform the Virchow elastic stain, deparaffinize the sections and hydrate to distilled water. The tissue section should be placed in Virchow elastic tissue stain for 1 hour. Then they should be washed in 2 changes of distilled water. The tissue should be differentiated microscopically in 2% ferric chloride until the elastic fibers are distinct and the background is colorless to light gray in color. If they are differentiated too far, the tissue should be restained. Then rinse in distilled water and put in sodium thiosulfate for 1 minute, then wash in running tap water for a 5-minute period. After this 5-minute period, the sections then need to be counterstained in Van Giesen for 1 minute, then differentiated at 95% alcohol. The tissue should then be dehydrated in absolute alcohol, cleared with xylene, then mounted with synthetic resin. We are going to look at photos of what the stain does look like, but let's first talk about the results of what they should look like when using this stain. So when stained with the Weerhoff elastic stain, elastic fibers should be blue-black to black in color. Nuclei will also be the same color. Collagen will be red, and other tissues in the section will be yellow. Mercury deposits, if present, do not need to be removed before staining because the staining solution will remove them. The photo on the left-hand side of the slide here shows a Weerhoff Van Giesen stained aortic wall, which is an elastic artery. The black lines on this section are the perforated laminae of elastic tissue. The yellow in this photo is the smooth muscle, so the smooth muscle cells, and the red color are the collagen fibers. The photo on the right-hand side of the slide is the same tissue, but just has been over-differentiated. So see how there is less black on the photo on the right-hand side? So that black are the elastic tissues. So the lack of the slightly grayed background and bright yellow muscle fiber staining is indicative of over-differentiation. The photo on the left-hand side of the slide shows a muscular artery cross-section that is stained with the Weerhoff Van Giesen stain. The black color here are the very fine elastic fibers. The yellow is the muscular tunic media, which is the middle layer of the blood vessel, and the collagen fibers are stained in this red color. The right-hand photograph is the skin section that is stained with Weerhoff Van Giesen stain. This is actually a bad staining example. Recall that the collagen fibers should be red when properly stained. So it's a yellow-orangish color in this photo. So this means that the picric acid used in the stain was not saturated. So this one should be restained with properly prepared stain. The next connective tissue stain is the aldehyde fusion elastic stain. So this is used to show changes in elastic fibers. In this stain, hydrochloric acid and perolaldehyde combine with basic fusion, which form an aldehyde fusion. And this has an affinity for elastic fibers. The preferred fixative is 10% neutral buffered formalin. Chromate fixatives cannot be used for this stain. Paraffin tissue section should be cut 4 to 5 microns in thickness, and the quality control tissue for this is the same for the Weerhoff stain, so aorta on an edge or a cross section of a larger artery. Skin can also be used as a quality control tissue for the stain. The reagents required for the aldehyde fusion elastic stain are as follows. Aldehyde fusion solution, which is prepared by combining 1 gram of parerosaniline, 200 milliliters of 70% alcohol, 2 milliliters of hydrochloric acid, and 2 milliliters of peraldehyde. Light green stock solution, which is prepared by combining 0.2 grams of light green SF, 100 milliliters of distilled water, and 0.2 milliliters of glacial acetic acid. And then lastly, light green working solution, which is prepared by combining 10 milliliters of light green stock solution with 50 milliliters of distilled water. To perform the aldehyde fusion elastic stain, deparaffinize the tissue and hydrate to 70% alcohol. Stain tissue sections with the aldehyde fusion solution for 10 to 40 minutes. After this step, rinse with 70% alcohol to remove any excess stain. Then wash with water and then check microscopically for elastic fiber stain. Then do a distilled water rinse. At this point, the tissue needs to be counterstained with light green working solution for 1 to 2 minutes, and after that step, dehydrated with 2 changes of 95% and absolute alcohol, clear with xylene, and then mount with synthetic resin. When stained with the aldehyde fusion elastic stain, elastic fibers will be deep blue to purple in color, and other tissues will be green. This photo shows a skin section that is stained with this stain. Elastic tissue here is stained a dark purple color, and this greenish, honestly, it kind of looks a bit blue in this photo, to be honest with you, but that is the epidermis and connective tissue of the dermis of the skin. The Russell modification of the Movat pennichrome stain is the next stain we'll be discussing. So this stain is used for the detection of mucin, elastic fibers, fibrin, muscle, and collagen. In this stain, Alcian blue stains acidic mucosubstances. Iron hematoxylin stains the elastic fibers if present, and they then are differentiated with ferric chloride. Any leftover iodine is removed by sodium thiosulfate. Acid fusion and crochet scarlet stain muscle, cytoplasm, ground substance, and collagen fibers. Then phosphotungstic acid removes the stain from collagen and ground substance, and acetic acid removes the phosphotungstic acid. Then the collagen is counterstained with an alcoholic safran. For this stain, tissue should be fixed in neutral buffered formalin or acetic formalin sublimate, and the paraffin section should be cut from 4 to 5 microns in thickness. Quality control tissue for the Russell modification of the Movat pentachrome stain are lung, skin, or colon tissue specimens. The reagents needed for the Russell modification of the Movat pentachrome stain are as follows. 1% Alcyon blue, which is prepared by combining a gram of Alcyon blue 8GS with 100 milliliters of distilled water and 1 milliliter of glacial acetic acid. Alkaline alcohol, which is prepared by combining 90 milliliters of 95% alcohol with 10 milliliters of ammonium hydroxide. Iodine iodide, which is prepared by combining 2 grams of iodine with 4 grams of potassium iodide and 100 milliliters of distilled water. 10% absolute alcoholic hematoxylin, which is prepared by combining 10 grams of hematoxylin with 100 milliliters of absolute alcohol. 10% ferric chloride. This is prepared by combining 10 grams of ferric chloride and 100 milliliters of distilled water. Hematoxylin solution, which is prepared by combining 25 milliliters of 10% absolute alcoholic hematoxylin with 25 milliliters of absolute alcohol, 25 milliliters of 10% ferric chloride, and 25 milliliters of iodine iodide. 2% ferric chloride, which is prepared by combining 10 milliliters of 10% ferric chloride with 40 milliliters of distilled water. 5% sodium thiosulfate, which is prepared by combining 5 grams of sodium thiosulfate with 100 grams of distilled water. Crochet scarlet acid fusion, and this is created by combining solution A and solution B. And solution A is prepared by combining 1 gram of the crochet scarlet with 99.5 milliliters of distilled water and 0.5 milliliters of glacial acetic acid. Solution B is prepared by combining 0.1 gram of acid fusion with 99.5 milliliters of distilled water and 0.5 milliliters of glacial acetic acid. So the working solution is combining 8 parts of that solution A with 2 parts of solution B. Then 5% phosphotungic acid solution, which is prepared by combining 5 grams of phosphotungic acid with 100 milliliters of distilled water. And then lastly, the last reagent here is the alcoholic safran, which is prepared by combining 6 grams of the safran deugatinase with 100 milliliters of absolute alcohol. So let's get into the procedure here. So to perform the Russell modification of the Movat pentachrome stain, the tissue should be deparaffinized and hydrated to distilled water. It then should be stained with Alcyon blue for 20 minutes and then washed in running tap water for 5 minutes. Tissue slides should then be put into alkaline alcohol for an hour and then washed in running tap water for 10 minutes, followed by a distilled water rinse. After this distilled water rinse is completed, it should be stained in hematoxylin solution for 15 minutes, and then followed by rinsing with several changes of distilled water. Then the tissue should be differentiated with 2% aqueous ferric chloride. This should be differentiated until the elastic fibers contrast sharply with the background under the microscope. We're going to continue this on the next slide here. So after the tissue section is differentiated appropriately with that 2% aqueous ferric chloride, it should then be rinsed in distilled water and then placed in sodium thiosulfate for 1 minute. After this 1 minute time interval, it should be washed in running tap water for 5 minutes, then rinsed in distilled water. The tissue should then be stained with the crochet scarlet acid fusion for 1.5 minutes. Following the staining step, the tissue section should be rinsed in several changes of distilled water, followed by a rinse in 0.5% acetic acid solution. Then, the tissue slides should be placed in 2 changes of 5% phosphotungic acid solution at 5 minutes each. So 2 changes at 5 minutes each, so at a 10-minute period. So following this, the tissue should be rinsed with 0.5% acetic acid solution. And we need to go on the next slide here. So following the rinse with the 0.5% acetic acid solution, the tissue slide should be rinsed in 3 changes of absolute alcohol. It then needs stain and alcoholic saffron solution for 15 minutes. After this 15-minute period, the tissue section needs to be rinsed in 3 changes of absolute alcohol, cleared, and 2 to 3 changes of xylene, and then mounted with a synthetic resin. After being stained with the Russell modification of the Movat pentachrome technique, both nuclei and elastic fibers in the tissue will be black in color. Collagen will be yellow. Ground substance and mucin will be blue in color. Fibrinoid and fibrin will be an intense red color, and muscle will be a regular red color. So the photo on the right-hand side of the slide shows a colon tissue section that is stained with the Russell modification of the Movat pentachrome stain. The mucin and the goblet cells are these blue structures here. So these here, all right, so blue, that's going to be the mucin and the goblet cells. The red stained elements, all right, red stain here, those are smooth muscle cells. The yellow stain elements, which are here, right, so those are collagen, and then all this black stuff here, black that can be seen here, those are the elastic fibers that are present within the tissue. There are 2 silver techniques for the staining of reticular fibers. These are the Gamori stain for reticular fibers and the Gordon and Sweet stain for reticular fibers. So before we talk about each of these individual stains, it's important to understand how the silver techniques work. They all have the same major steps to staining. So the first step is oxidation. So this is the oxidation of glycol groups in reticulin fiber to aldehydes. The most commonly used oxidizers are phosphomylic acid, potassium permanganate, and periodic acid. Sensitization is frequently a metallic impregnation step. So this means that there are metallic salts deposited on or around the part of the tissue that is supposed to be demonstrated in the stain. Excuse me. This sensitization material is then replaced with silver. Silver impregnation occurs when treating the tissue with any ammoniacal or diamine silver complex. The reduction step uses formaldehyde to reduce any leftover silver diamine ions and then reduces them to a metallic silver. Toning is when the bound metallic silver is treated with gold chloride. This changes the metallic silver to metallic gold. Then the unreduced silver is removed by treating the tissue sample with sodium thiosulfate. The last step is counterstaining. So whether or not this step is performed is dependent on what type of tissue that is being stained. So these steps all occur with silver techniques for staining reticular fibers. So now let's talk about those 2 silver techniques. The Gamori stain for reticular fibers is a silver technique that helps to show the reticulin present in tissue sections. This is particularly helpful in the diagnosis of certain tumor types. For this stain, potassium permanganate oxidizes hexose sugars in reticulin fibers to aldehydes. The excess is removed by the potassium metabisulfite. Ferric ammonium is the sensitizer in this stain, and formalin is used to reduce silver to its metallic form. Toning occurs with gold chloride, and sodium thiosulfate is used to remove unreacted silver from the stain. Paraffin sections need to be cut to 4 to 5 microns in thickness and fixed with 10% neutral buffered formalin. For the Gamori stain for reticular fibers, quality control tissue should be tissue from the liver. The reagents for the Gamori stain for reticular fibers are as follows, and as a note here we have 2 slides full of reagents. 10% silver nitrate solution, which is prepared by combining 10 grams of silver nitrate with 100 milliliters of distilled water. 10% potassium hydroxide solution, which is prepared by combining 10 grams of potassium hydroxide with 100 milliliters of distilled water. Ammoniacal silver solution, 0.5% potassium permanganate solution, which is prepared by combining 2.5 grams of potassium permanganate with 500 milliliters of distilled water. 2% potassium metabisulfite solution, which is prepared by combining 10 grams of that potassium metabisulfate with 500 milliliters of distilled water. 2% ferric ammonium sulfide solution, which is prepared by combining 10 grams of ferric ammonium sulfate with 500 milliliters of distilled water. Let's go to the next page here. We've got more reagents. Formalin solution is also required for this stain. This is prepared by combining 10 milliliters of 37 to 40% formaldehyde with 40 milliliters of distilled water. 0.2% gold chloride solution, which is prepared by combining 10 milliliters of stock gold chloride solution with 40 milliliters of distilled water. 2% sodium thiosulfate solution, which is prepared by combining 10 grams of sodium thiosulfate with 500 milliliters of distilled water. And then lastly, nuclear fast red solution, which is prepared by combining 0.5 grams of nuclear fast red with 25 grams of aluminum sulfate and 500 milliliters of distilled water. To perform the Gamori stain for reticular fibers, the tissue should be deparaffinized and hydrated to distilled water. The section should be oxidized with 0.5% potassium permanganate for 1 minute. Then, the tissue should be rinsed in tap water for 2 minutes. The tissue should then be differentiated with 2% potassium metabisulfate for 1 minute, followed by a 2-minute wash in tap water. The tissue should then be sensitized with 2% ferric ammonium sulfate for 1 minute. And after this sensitization, the section should be washed with tap water for 2 minutes, then rinsed in 2 changes of distilled water at 30 seconds each per those changes. After these rinses, the tissue needs to be impregnated with silver solution for 1 minute, then rinsed again in distilled water for 20 seconds. So let's go to the next page here. After the 20-second rinse in distilled water, the tissue section needs to be reduced in formalin solution for 3 minutes. Following this reduction, washed in tap water for a 3-minute period. Next, the tissue needs to be toned in 0.2% gold chloride solution for 10 minutes, then rinsed in distilled water. The tissue section then needs to be placed in 2% potassium metabisulfite for 1 minute, and then fixed in 2% sodium thiosulfate for 1 minute. After these steps, the tissue needs to be washed in tap water for 2 minutes, then counterstained with nuclear fast red for 5 minutes if needed. Of course, this is an optional step, the counterstaining. Lastly, the tissue section will need to be dehydrated with 95% and absolute alcohol, cleared with xylene, and then mounted with a synthetic resin. When stained with the Gamori stain for reticular fibers, the reticulin will be black in color and collagen will be a taupe color. The photo on the left-hand side of the slide shows a liver tissue section that is stained with the Gamori reticulin stain, and then counterstained with nuclear fast red solution. So this one on the left-hand side is 10x objective on the microscope. Now the photo on the right-hand side shows the same section under the 40x objective. The reticulin fibers in black are easily visible in both of these images. So we're talking about this black stuff here, right? So all of that, oh goodness, I can't draw arrows. All this black on both of these slides here, those are the reticulin fibers. So the difference between this one here and this one here is this one is 10x objective and this one is 40x. It's the exact same tissue section, just looking at it different objectives. Now the Gordon sweet stain for reticular fibers is the second silver technique for reticular fibers. In this staining technique, the tissue is oxidized with potassium permanganate. Any excess of this oxidizer is removed with oxalic acid. Ferric ammonium sulfate is the sensitizer in this reaction. After impregnation of the tissue, formalin reduces the silver to its metallic visible form. Any excess silver is removed with sodium thiosulfate. And of course, counterstaining is optional. Paraffin sections should be cut from 4 to 5 microns in thickness and fixed in 10% neutral buffered formalin. Just like with the Gamori stain for reticular fibers, the quality control should be liver tissue. The reagents needed for the Gordon and Sweet stain for reticular fibers are as follows. 10% silver nitrate, which is prepared by combining 10 grams of silver nitrate and 100 milliliters of distilled water. 3% sodium hydroxide, which is prepared by combining 3 grams of sodium hydroxide with 100 milliliters of water. Actually, I probably should have labeled that as sodium hydroxide solution. That probably would make more sense. Ammoniacal silver solution, 1% potassium permanganate solution, which is prepared by combining 1 gram of potassium permanganate with 100 milliliters of distilled water. 1% oxalic acid solution, which is prepared by combining 1 gram of oxalic acid with 100 milliliters of distilled water. 2.5% ferric ammonium sulfate solution, which is prepared by combining a lot of things. 2.5 grams of ferric ammonium sulfate with 100 milliliters of distilled water. Formalin solution, which is prepared by combining 37 to 40% formaldehyde and 90 milliliters of distilled water. 0.2% gold chloride solution, which is prepared by combining 10 milliliters of 1% stock gold chloride solution and 40 milliliters of distilled water. Let's go to the next slide here with a continuation of the reagents needed for this stain. You also need 5% sodium thiosulfate solution, which is prepared by combining 5 grams of sodium thiosulfate and 100 milliliters of distilled water. Nuclear fast red solution, of course this is an optional step. This is prepared by combining 0.5 grams of nuclear fast red with 25 grams of aluminum sulfate and 500 milliliters of distilled water. All right, so to perform the Gordon and Sweet stain for reticular fibers, tissue should be deparaffinized and hydrated to distilled water. The section should be oxidized in 1% potassium permanganate, 1% potassium permanganate solution for 5 minutes, and then rinse with tap water for 2 minutes. After this tap water rinse, the tissue should be bleached in 1% oxalic acid. It should be bleached for 2 minutes or until it becomes colorless. Then it should be washed in tap water for 2 minutes. Then sensitize with 2.5% ferric ammonium sulfate for 15 minutes and then washed in several changes of distilled water. After the several changes of distilled water wash, the tissue section should be impregnated with silver solution for 2 minutes, then rinsed in distilled water. Let's go to the next page here because the procedure still is too long for one slide. So after this distilled water rinse, the tissue should be reduced in formalin solution for 2 minutes, then washed in tap water for 3 minutes. After this wash, the tissue should be placed in 5% sodium thiosulfate for 1 minute, then rinsed again in distilled water. After this rinsing step, tone in 0.2% gold chloride solution for 10 minutes, then wash in tap water for 2 minutes. 10 minutes, I believe. So sorry, I think I said that incorrectly. So 0.2% gold chloride solution for 10 minutes, then wash in tap water for 2 minutes. At this point, if a counterstain is indicated, counterstain with nuclear fast red solution for 5 minutes. And again, this counterstaining is an optional step. The tissue then needs to be dehydrated in 2 changes of 95% and absolute alcohol, cleared with xylene, and then mounted with the synthetic resin. When stained with a Gordon and Sweet stain, reticulin should be black in color. This photo on the left is a liver tissue section that has been stained with a Gordon and Sweet stain. There is no counterstain used here in this photo. All this black that is seen are the reticular fibers that line the sinusoids in the liver tissue. Now the photo on the right hand side is an example of a poorly performed Gordon and Sweet staining technique. And as you can see, the reticulin pattern, so the reticulin pattern on this left hand side is, see all this black here, right? Those are the reticular fibers. You can't see it on here. It's just obscured by the counterstain. Too much of that nuclear fast red, so it's just overstained with that counterstain. So this on the right hand side is an unacceptable stain and it needs to be repeated. The Mallory PTAH technique for cross striations in fibrin and the PTAH without mercuric solution are stains for muscles. And as a note, PTAH stands for phosphotungic acid hematoxylin technique. So the Mallory PTAH technique for cross striations and fibrin stain is for the demonstration of muscle cross striations and fibrin. And this is important because cross striations are a feature of rhabdomyosarcomas or striated muscle tumors. A rhabdomyosarcoma is a type of cancer that occurs in the soft tissue. Nemelin rods also show up with this stain, and these Nemelin rods are present in certain skeletal muscle diseases. So this stain is referred to as a polychrome stain because it gives two prominent colors. So within this stain, tungsten from the phosphotunxic acid binds hematine, and this binding produces a blue-colored lake. And this lake stains specific tissue parts blue, and the phosphotunxic acid stains components red, brown, in color. So two different colors here. The blue and then the red, brown in color. So that's why it's called a polychrome stain. So paraffin sections should be cut from 4 to 6 microns in thickness, and the preferred fixative is Zinker solution, although 10% neutral buffered formalin is acceptable. Quality control tissues for the Mallory PTAH technique are skeletal muscle or cardiac muscle for the demonstration of cross striations. Any tissue that has fibrin can be used as a control for fibrin demonstration, and a cerebral cortex tissue can be used as a quality control if the demonstration of glial fibers is needed. So glial cells are cells that help support neurons within the central nervous system. The reagents needed for the Mallory PTAH technique are as follows, and as I'm doing this lecture, I'm realizing you all can read how to prepare these reagents, so I'm not going to list how to prepare them, just going to list the reagents that are needed. So the PTAH solution is required: gram iodine, 5% sodium thiosulfate, 0.25% potassium permanganate, and 5% oxalic acid solution are required. To perform the Mallory PTAH technique, tissue needs to be deparaffinized and hydrated to distilled water. If the tissue is formalin fixed, mordant in Zinker with 5% acetic acid overnight at room temperature, then rinse with tap water. Then place in gram iodine for 15 minutes, then rinse in tap water. After this step, place the tissue section in 5% aqueous sodium thiosulfate for 3 minutes, then wash in tap water for a 10-minute period. After this 10-minute period, place the tissue sections in 0.25% potassium permanganate for 5 minutes. After this 5-minute time interval, rinse it in tap water. Then place the tissue in 5% oxalic acid for 1 minute. And after this 1-minute time interval, wash with running tap water for 10 minutes. After that 10-minute period, place in the PTAH solution overnight, keeping it at room temperature. The tissue then needs to be dehydrated rapidly with 2 changes of 95% and absolute alcohols, cleared with xylene, and then mounted with a synthetic resin. When stained with the Mallory PTAH technique, cross striations, fibrin, and nuclei will be blue in color. Collagen will be red-brown. So the photo on the left-hand side of this slide here shows a Zinker-fixed skeletal muscle that has been stained with the Mallory PTAH technique. The photo on the right-hand side of the slide shows the same thing, just on a very highly magnified view. So this stain is very rarely used today for the identification of cross striation in tumors that are thought to be rhabdomyosarcomas. Immunoenzyme staining is now the preferred method for this. So this photo shows a formalin-fixed skeletal muscle that is fixed with the Mallory PTAH technique. The uneven staining here is visible, and this is common with formalin-fixed tissue for this technique. So this is why the Zinker solution is preferred for this stain type. The PTAH stain without mercuric solutions does the same as the Mallory PTAH technique and has the same requirements. It just doesn't use a mercuric solution. So the reagents required for this stain are acidic dichromate solution, acidic potassium permanganate solution, and 1% oxalic acid. For this procedure, the tissue should be deparaffinized and hydrated to distilled water. Place in acidic dichromate solution for 30 minutes and then wash with tap water. Then it should be placed in acidic permanganate solution for 1 minute, then washed with tap water again. The tissue section that needs to be bleached or the tissue section does need to be bleached in 1% oxalic acid solution, then rinsed again with tap water. It should be placed in PTAH solution overnight. And after the soak in that PTAH solution, it should be dehydrated rapidly with 95% in absolute alcohols, cleared with xylene, and then mounted. The periodic acid methenamine silver microwave procedure for basement membranes is a stain for, well, you guessed it, basement membranes. So this stain is used to highlight the basement membrane. So now recall from super far beginning part of this lecture that the basement membrane or basal lamina separates the epithelial cell layer from the underlying connective tissue. In this staining technique, methenamine silver oxidizes the carbohydrates present in the tissue to aldehydes, and this causes a reduction showing visible metallic silver. The toning in this procedure is done with gold chloride, and unreduced silver is removed with sodium thiosulfate. Paraffin sections need to be cut at 2 microns in thickness and fixed in 10% neutral buffered formalin. No mercury-containing fixative should be used for this staining procedure. If staining kidney tissue, this has an internal control, but no other control slide is necessary for this particular staining technique. The reagents needed for the periodic acid methenamine silver microwave procedure are stock methenamine silver, 5% borax solution, working methenamine silver solution, 1% periodic acid solution, 0.02% gold chloride solution, stock light green solution, and working light green solution. For the periodic acid methenamine silver microwave technique, 5 slides must be stained. If there are not 5 slides of tissue, blank slides need to be used to ensure that 5 slides are all being stained together. The tissue needs to be deparaffinized and hydrated to distilled water. The section should be placed in 1% periodic acid solution for 15 minutes at room temperature, then rinsed in distilled water. 5 tissue slides should be placed in a plastic Copeland jar that contains 50 milliliters of the methenamine working solution. Its cap should be put on loosely and put in the microwave oven. The jar should be put in the microwave oven, not just the cap. Another plastic Copeland jar that contains 50 milliliters of distilled water with a loose cap should also be placed in the oven. Then they need to be microwave full power for 70 seconds. Remove both the Copeland jars, mix the contents with a Pasteur pipette, and let it stand. The slides do need to be checked frequently. However, 15 to 20 minutes is what it takes usually until the desired staining is achieved. After this step, the 5 slides need to be rinsed with heated distilled water. Then they will need to be toned in 0.02% gold chloride for 30 seconds and rinsed with distilled water. After this distilled water rinse, the slide should be treated with 2% sodium thiosulfate for 1 minute. Following this treatment, wash with tap water, and let's go to the next slide here. So after that tap water wash, the slide should be counterstained with a working light green solution for 1.5 minutes. Then they should be dehydrated with 2 changes of 95% and absolute alcohols, cleared with xylene, and then mounted with synthetic resin. When stained with the periodic acid methenamine silver microwave procedure technique, basement membrane should be black in color and the background should be green. The photo on the far left-hand side of the slide here shows a kidney glomerulus that is stained with this. So this is a great example, and Dr. Carson describes that a good stain will show the basement membrane like it was drawn with ink. The middle photo of the slide shows another kidney glomerulus stained with this stain. This has been understained. So the photo on the right-hand side of the slide, so the far right-hand side, shows an overstained kidney glomerulus. So this is also too thick of a section to be stained. So recall that the tissue should be cut at 2 microns for optimum staining using this method. There are 3 different staining techniques for lipids that we're going to be discussing in this lecture. These are the oil red O method for neutral fats technique, Sudan black B and propylene glycol technique, and the osmium tetroxide paraffin procedure for fat technique. The oil red O method for neutral fats is a technique used for the demonstration of neutral lipids within frozen tissue sections. So the detection of neutral fats are important, as fat that is present in abnormal locations can indicate that there was a fatty embolus that has developed usually by a fracture of the bone or other type of crush injury. So also tumors that come from fatty cells, these are called liposarcomas, can help to be detected with this type of stain. So frozen sections need to be cut to 10 microns in thickness. 10% neutral buffered formalin or calcium formol should be the fixatives used. Alcoholic fixatives need to be avoided as alcohol can dissolve lipids. And of course, we are looking for lipids within the stain, so we don't want them to be dissolved here. So most tissues have some fat in them, so a control is usually not indicated to ensure that the stain is working properly. The reagents needed for the oil red O method are oil red O stock solution and oil red O working solution. So this procedure for this is pretty simple in comparison to some of the other stains that we have talked about in this lecture so far. So the frozen sections need to be cut and fixed in 40% formaldehyde for 1 minute, then washed in tap water. Excess water needs to be blotted off, and just as a note here, if the tissue has already been fixed, there's no need to refix it. Then the section needs to be stained with oil red O for 10 minutes, then washed with tap water. Then it needs to be stained with Harris hematoxylin with acetic acid for 1 minute. Then washed again with tap water. The sections then need to be blued in ammonia water, then washed once again with tap water. So at this point, the section needs to be mounted with aqueous mounting medium, with the cover slip edges being sealed with a fingernail polish. When stained with the oil red O method, nuclei will stain blue and fat will be stained an intense red color. The photo on the left-hand side of the screen shows a frozen section of a fatty liver tissue that is stained with the staining technique. So there are nuclei present in blue here, and then of course the intense red shows the fat. The photo on the right-hand side shows the same tissue type, but showing an issue during cover slipping. So air bubbles were chased out from under the cover slip by mashing the cover slip during the mounting step, and this caused issues. So because fat is relatively liquid, it's very important to cover slip properly. The Sudan black B and propylene glycol method is another staining method for neutral lipids present in the tissue. So this is actually the most sensitive dye for lipids. So we also use Sudan black B and medical laboratory technology. It's used to determine the difference between granulocytic precursor cells from lymphocytic and monocytic precursor cells. So tissue should be fixed in 10% neutral buffered formalin or post-fix in calcium formalin. No alcoholic fixative should be used, as we already know that alcohol dissolves lipids, and we're looking for lipids, right? So we don't want it to be dissolved. So frozen sections need to be cut to 10 microns in thickness. Paraffin sections are not able to be used for this because the dehydrating and clearing agents to be used for that dissolve fats. Most tissues have some fat present, so no external control tissue is indicated for this staining method. The reagents that are needed for the Sudan black B and propylene glycol are calcium formalin and Sudan black B staining solution. And of course, you can read on this slide here how to prepare these reagents. To stain the tissue with the Sudan black B and propylene glycol method, fixed and rinsed cryostat sections need to be placed in 100% propylene glycol for 2 changes at 15 minutes. After this step, the tissue should be stained with the Sudan black B for 10 minutes, then differentiated with 85% propylene glycol. At this point, the tissue needs to be washed in distilled water. The tissue then needs to be counterstained with nuclear fast red for 1 to 5 minutes. After that 1 to 5 minute period with the nuclear fast red, it then needs to be washed with several changes of distilled water, then mounted with an aqueous mounting medium. When stained with the Sudan black B and propylene glycol stain, fat will be blue-black in color and nuclei will be red. So this photo on the right-hand side here, this slide shows a fatty liver frozen section. All this black here is fat that is present within the tissue. So all of this here, right? All of this is going to be that black stuff, right? So that's all going to be the fat within the tissue. The osmium tetroxide paraffin procedure for fat allows for the tissue to be embedded with paraffin. So in this stain, the osmium tetroxide combines with the fat and blackens it. Tissue should be fixed with 10% neutral buffered formalin. The tissue block should be wetted, then processed beginning in 70% alcohol. It's imperative that the tissue not be thicker than 2 millimeters or the osmium will be unable to penetrate into the tissue. Most tissues have fat present, so no external quality control tissue is indicated here. The reagents needed for this technique are 1% osmium tetroxide solution and 0.5% periodic acid solution. To be prepared using the osmium tetroxide paraffin procedure, trim the tissue to 2 millimeters thick and wash it in running tap water for 30 minutes. It then needs to be rinsed in distilled water. The tissue should be then placed in 5 milliliters of osmium tetroxide solution in a container with a cap. Cap the container, then leave for 1 to 2 hours under the hood. And this solution should be periodically agitated during this 1 or 2 hour period. The tissue type is going to determine how long it should be left, either 1 or 2 hours. So heart tissue needs 2 hours, aorta and lung tissue needs 1 hour. So after this designated length of time, the tissue needs to be rinsed with 2 changes of distilled water at 15 minutes each, then differentiated in 0.5% periodic acid for 30 minutes. It's important to agitate this periodically throughout that 30-minute period. So after that time is up, wash it with tap water for 30 minutes. And after this 30-minute period, the tissue should be processed routinely, starting with 70% alcohol and embedded routinely. The paraffin sections are cut 4 to 5 microns in thickness, picked up on slides, and then routinely dried. The tissue then can be deparaffinized and hydrated. It can be stained with a routine H&E stain or with any special stain as desired. Masson trichrome is a great counterstain for osmium fixed tissue sections. After staining, the section should be dehydrated, cleared, and then mounted with synthetic resin. When stained with the osmium tetroxide paraffin procedure, fat will be black in color. And depending on the method used, tissues can be different colors. So this photo here shows a liver section that was fixed with osmium tetroxide, and then it was stained with the Masson trichrome procedure. So the fat here is shown as these black spots throughout the tissue. So these here, all these black spots are going to be the fat. There are 2 staining techniques for connective tissue cells. So these are toluidine blue stain for mast cells and methyl green pyronin Y. So we've discussed this methyl green pyronin Y in the nuclear and cytoplasmic staining lecture. So I won't be discussing in this lecture other than that mast cells stain an orange to red color with this particular stain. So if you want to know more about that particular stain, please check out my nuclear and cytoplasmic staining lecture. It's available within the playlist of this histopathology lecture series. Toluidine blue is a stain for the detection of mast cells present in tissue samples. So mast cells are stained metachromatically with this stain, meaning that they stain a different color than the entire rest of the tissue. Tissue should be fixed in 10% neutral buffered formalin and paraffin sections cut to 4 to 5 microns in thickness. Quality control for this stain should be any tissue that contains known mast cells. Reagents required for this stain are just the toluidine blue solution, and this is prepared by combining 0.1 grams of toluidine blue with 100 milliliters of distilled water. To perform the toluidine blue for mast cell stain, deparaffinize the tissue and hydrate to distilled water. Then stain with toluidine blue solution for 10 minutes. After this 10-minute staining period, rinse with distilled water. Then quickly dehydrate with 95% in absolute alcohols, clear with xylene, and then mount with synthetic resin. After successfully staining with toluidine blue, mast cells will be a deep violet in color, and the background will be blue. This photo shows a skin section from a mastocytoma, actually from a canine patient. So a mastocytoma is a tumor of mast cells and can occur in humans and other animals, like in this dog. This section shows metachromatically stained mast cells that are present. Alright, so that's it for this lecture. So if this video helped you out, please give it a like. Please subscribe and share my channel for more educational and laboratory content. And as always, if you have any questions about this lecture, please feel free to leave them in the comments section. I would be happy to answer them for you. All right, until next time.